Welcome to Chapter 7, Part 5. We're going to learn some about alternative pathways, including fermentation, and what happens in different organisms, and including ourselves, with fermentation. So fermentation is a type of anaerobic respiration, but we don't really call it anaerobic respiration because we're not going through chemiosmosis. In fact, for the processes of fermentation that we're going to talk about, it doesn't involve oxygen. And in fact, there are times in our own bodies where we don't have enough oxygen. So instead, we're going to make ATP without oxygen, which means we have to cut out the electron transport chain and we have to cut out oxidative phosphorylation. The only way that we're going to make ATP is through substrate-level phosphorylation. And no matter which one we talk about, which type of fermentation we talk about, we're going to be starting with glycolysis. Again, we're not going to talk too much about the anaerobic respiration part of this, but both fermentation and anaerobic respiration, there's no oxygen involved. With anaerobic respiration, there might be some other molecule that acts as an electron acceptor instead of oxygen. For example, sulfate is used in the final electron acceptor instead of oxygen in some organisms, like botulism. On the other hand, fermentation is a bypass which allows continuous production of ATP in substrate-level phosphorylation in glycolysis. So there are two main kinds of fermentation. There's alcohol fermentation, which we see right here, and there's lactic acid fermentation, which we see right here. Both alcohol fermentation and lactic acid fermentation tell you what the final step is, the final product is in their chemical process. Alcohol fermentation has alcohol as a byproduct, and lactic acid fermentation has lactic acid as a byproduct. Both of these, importantly, consist of glycolysis plus a second set of reactions that cause NAD+ to be regenerated and used again in glycolysis. So remember NAD is used up in glycolysis. So if and also remember that NADH would be normally used in the electron transport chain, chemiosmosis, oxidative phosphorylation, but we don't have any oxygen around. And so if there's no oxygen around, we're not going to be using up NADH anyway. So we're going to take the NADH that we made in glycolysis and we're going to turn it back into NAD+ in the process of fermentation. Let's start out here with alcohol fermentation. With alcohol fermentation, we've got a couple of steps here. Let's focus on glycolysis for just a second and remember that 2 ATPs are made in glycolysis and our end product there is pyruvate, right? So what we're doing here is we're going to, and also remember that NAD+ is made into NADH. So what we're doing here is we're going to regenerate NAD+, and as a byproduct, we're making ethanol. We're making 2 ethanols for every glucose molecule that enters this process. Carbon dioxide is released from the pyruvate here, and we make carbon dioxide as a byproduct. This is why beer and champagne, things like that, have carbon dioxide in them, because it's a byproduct of the process. Acetaldehyde is then turned into the ethanol, and it's the alcohol that people like to drink when they're drinking alcohol. It's the thing that gets you drunk. Alcohol fermentation by yeast is used in brewing and beer. It's also used in wine making, and interestingly, and you might not be aware of this, but it's also used in baking. When you bake bread with yeast, you get carbon dioxide, and that carbon dioxide causes the bread to puff up, and that's why the bread is all puffy. The alcohol, when it's baked, is all baked off. So we don't, when we eat bread, we don't really, we aren't imbibing alcohol, but it is one of the byproducts. In lactic acid fermentation, it's the same basic idea, except for yeast don't do lactic acid fermentation. We do lactic acid fermentation. So if we're going through glycolysis, we're going to make our 2 ATPs, and that's good. We can use those for energy. But then we're making our pyruvate. And we have all this NADH around that we don't need because we don't have any oxygen. Think about times where you've worked out so hard that you're lifting weights. That's a good example. And then your muscles start to burn and ache. That's because you're running out of oxygen. You can still move, but you're bypassing that oxidative phosphorylation with our NADH. No oxygen, not enough oxygen around. So we're going to regenerate our NAD+ by turning pyruvate into lactate. And lactate is also known as lactic acid. So again, the processes are very, very similar. It's just with alcohol fermentation, you have carbon dioxide and ethanol as a byproduct, but most importantly, you're regenerating your NAD+ so that you can go through glycolysis again. Same thing for lactic acid fermentation. You've got your byproduct. This can cause muscle aches and pains, but very importantly, we're regenerating our NAD+ so that we can go through glycolysis again. Human muscle cells absolutely use lactic acid fermentation to generate ATP when there's not enough oxygen around. Lactic acid fermentation is also used by some fungi and bacteria, and we take advantage of that to make cheese and yogurt. Yeast and bacteria are also responsible for making chocolate. So these are some pretty important chemical processes in our lives. All 3 processes, fermentation, anaerobic respiration, which again we haven't talked about too much, and aerobic respiration, all 3 processes use glycolysis. That's just it. And remember, only 2 ATPs are made from glycolysis. So to oxidize glucose and other organic fuels like fats and so forth, we're making pyruvate is the whole thing here. In all 3 of these, NAD+ is the oxidizing agent that accepts electrons from food during glycolysis. That's important. And in all 3 of these processes, ATP is made by substrate-level phosphorylation. When the main differences here are that what happens to the NADH. And in fermentation, the final electron acceptor is an organic molecule like pyruvate or acetaldehyde. In cellular respiration, the NADH is transferring its hydrogens and electrons through to oxygen. With anaerobic respiration, instead of oxygen, it might be to sulfur. Cellular respiration is going to produce about 32 ATPs per glucose, and fermentation is only going to produce 2 ATPs per glucose. Some organisms absolutely cannot stand oxygen, and they actually get killed in the presence of oxygen. So obligate, that means they're obliged to, they must. Obligate anaerobes only use fermentation or anaerobic respiration and can't survive in the presence of oxygen. Facultative anaerobes. Facultative means, yeah, maybe we can do it, maybe we can't. Sometimes we do, sometimes we don't. Facultative anaerobes, like yeast and lots of bacteria, can use either cellular respiration when there's oxygen around, or fermentation when oxygen is absent. It's really interesting about how glycolysis evolved, because this is the most common metabolic pathway. So it evolved very early on in the history of life. In early Earth, there was not a lot of oxygen available in the atmosphere. We think that the atmosphere of the early Earth was composed of gases similar to those found in the erupting volcanoes that we see today. So volcano eruptions give lots of water vapor, carbon dioxide, nitrogen, and other gases, but not a lot of oxygen. That's compared to today, where we have about 21% oxygen and 78% nitrogen. So we do think that early life did not have oxygen around, so didn't have oxidative phosphorylation. Instead, they relied strictly on glycolysis and fermentation. And so glycolysis, because it occurs in the cytosol, we think that it evolved before the mitochondria evolved. We also think that oxygen didn't become abundant in the atmosphere until after photosynthesis was invented. We'll talk more about that in the next chapter. I've been mentioning throughout these lectures that glycolysis we think about as using glucose. That's a major metabolic pathway, but it's not the only metabolic pathway. Let's take a look at the way some other things get used up in other parts of the food that we get used up in the process of making ATP. So there are catabolic pathways. Remember, catabolism is to break down, and it funnels all kinds of electrons from all kinds of food that we eat. For example, we can take carbohydrates and break them down into simple sugars like glucose or fructose. And those can go through glycolysis and make acetyl-CoA. Same thing for proteins. We can break down proteins into amino acids, and those amino acids can be eventually formed into acetyl-CoA. And the same thing for fats. So fats can be broken down into fatty acids and glycerol, and those can be made into acetyl-CoA. And then we can run the citric acid cycle. We'll look at that in a little bit more detail in a second. For now, we're just going to get an overview of the versatility of this process of breaking down molecules. So again, fats are digested into glycerol and fatty acids, and they're going to be broken down into 2 carbon fragments by this process called beta oxidation. And that 2 carbon molecule, carbon fragments, those are going to give us acetyl-CoA. If you take 1 gram of fat, it can produce almost twice as much ATP as 1 gram of carbohydrate, which is why it has so many calories to it, right? All right, let's take a closer look at our catabolism and look exactly where our different parts are going to enter the glycolysis, pyruvate, acetyl-CoA, those kinds of things. So for the most part, proteins are broken down into their amino acids. And amino acids have that amine group and that acid group, right? Well, the amine group is going to be discarded. And the acid groups and other parts of the amino acid, like the R groups, are going to enter the glycolysis as pyruvate. Carbohydrates like starch and things along those lines, glycogen, those can be broken down into their component sugars, and specifically here glucose, and glucose is going to enter glycolysis. Sometimes if you're breaking down sucrose is made up of 2 sugars, glucose and fructose, and so glucose would enter glycolysis as glucose, and fructose would enter as fructose 6-phosphate. Fats can be broken down as well. Glycerol is going to enter as glyceraldehyde 3-phosphate, and the fatty acids are going to enter, as we said before, as acetyl-CoA. Some amino acids will have parts of their structures entering directly as acetyl-CoA. But eventually, you got to make the acetyl-CoA in order to enter the citric acid cycle. Now let's think about how our bodies can use these same processes to build molecules, and those are anabolic pathways. Food is used to provide small molecules that are broken down, but then those small molecules are going to be used to build up something else. So with our catabolism, we can make smaller molecules to make energy, but we can bypass that energy. We don't have to send them to make ATP. We can use them to build larger molecules, specifically the ones that we need for our body's needs. So some molecules that we eat, that we need, can come directly from food. Others have to, they're products of glycolysis or the citric acid cycle. So just a really quick review here. I know it's been a lot, but if you look at the summary, it's not so bad. Glycolysis, your inputs are glucose, your outputs are pyruvate, 2 ADP, and 2 NADHs. With the citric acid cycle, our inputs are 2 pyruvates, which get changed into 2 acetyl-CoAs. And remember that we have 2 oxaloacetates here that are going to join up with the acetyl-CoA. Churning twice, you're going to get 2 ATP, 8 NADHs. Remember, we get some from pyruvate. 6 carbon dioxides, and that's every single carbon in glucose. Remember, it's C6H12O6. So we've accounted for all of our carbons now in carbon dioxide, and 2 FADH2s. The electron transport chain is important because it's transferring our electrons along this chain here. Eventually, the final electron, the acceptor, is oxygen, and that's going to join up with a couple of protons. But importantly, the movement of those electrons is pushing protons into the intermembrane space of the mitochondria, and that intermembrane space is then going to be filled with protons, which then are going to be allowed to move down through ATP synthase into the matrix, and that's what makes ATP, the vast majority of ATP. I hope that you've learned a lot from this chapter. There is a lot to learn, so feel free again to watch those animations or listen to my lecture again, and let me know if you have any questions.